Now the intermediate filaments. Intermediate filaments are intermediate, as their name indicates. They are intermediate between the microfilaments and the microtubules. They are about 10 nanometer in diameter, which is the greater than that of microfilaments and lesser than the microtubules. Now, some of the biochemical aspects of intermediate filaments. Their structural unit is a protofibril. Each protofibril is an antiparallel tetramer of two rod-like dimers. We can understand it by looking in the image. uh, they are the two monomers. These are the two monomers. Okay. Now these are the dimers. ठीक है, अब एंटी पैरेलल कैसे है कि यहाँ कार्बोक्सिल टर्मिनल है, यहाँ अमाइनो टर्मिनल है इस डायमर का यहाँ अमाइनो टर्मिनल है और यहाँ कार्बोक्सिल टर्मिनल है. ठीक है, so they are not parallel, they are not coinciding with each other. Carbon is not with carbon, instead it is with nitrogen. So they are anti-parallel tetramer of two rod-like dimers. ठीक है, and four protofibrils unite to form a cable of intermediate filament. Four protofibrils, each is being formed by the joining of the two dimers to form an anti-parallel tetramer, and the protofibril is made by the uniting of the intermediate filament is made by uniting of four protofibrils. Now the intermediate filaments are of 6 types: keratin filaments, Desmin filaments, Vimentin filaments, Neurofilaments, Glial filaments, and laments. We will discuss them one by one. Now keratin filaments. They can be acidic or basic in the human body. The keratin filaments or the cytokeratins are abundant in the epithelial cells, mostly in that of skin, the epidermis of skin. ठीक है, epidermis में बहुत होते हैं keratin filaments. So the keratin filaments, they form bundles which are called as tonofibrils, which are attached to the cell-to-cell junctions, the cell-to-cell connections, the cell-to-cell appositions. ठीक है, they help in the process of keratinization. That is, they form an outer impermeable layer on the epidermis of the skin which is resistant to dehydration. ठीक है, on our skin there is an outer impermeable layer which resists the dehydration. It is made up of keratin filaments. They help in stabilizing the cell shape, they maintain the cell junctions and attachments, and they prevent cellular injuries. The keratin filaments are present in skin, nails, horns, feathers, etc. We can look in this image that keratin filaments are bundled to form the tonofibrils, and here are the cell junctions, and they are attaching with the cell junction here also. Vimentin filaments, they are present in the cells derived from the mesenchyme. Now what is mesenchyme? Mesenchyme is the embryonic connective tissue. Okay, which the cells derived from the mesenchymes are fibroblasts, macrophages, etc. And the Vimentin filaments can be randomly distributed or they can be bundled into rods and other structures. Now look at the image. It is a special histochemical image. The yellowish green appearance of the fibers represent the Vimentin filaments. Here is a nucleus and this whole is the mesenchymal cell, and the yellowish green appearance is the Vimentin filament. Desmin filament. They are present in muscular tissue. ठीक है, in smooth muscles they help in the uniform contraction, and in the skeletal muscles and the cardiac muscles, which are together called as the striated muscles, they help in attaching of the Z discs, which are the borders of the sarcomere, to the cell membrane. ठीक है, in striated muscles they help in attaching the Z discs of the peripheral sarcomeres to the cell membrane. You can look in this image. These are the Z discs. They are attaching to the cell membrane with the help of Desmin filaments. Okay, and here is the conductile apparatus. Neurofilaments, they are found in neurons for their structural supports. Here is the neuron, and here inside it, in the core of it, there is the neurofilament. Glial filaments, they are present in neuroglia, which are the supporting cells of the nervous tissue, the astrocytes and the Schwann cells and the other cells, and the oligodendrocytes as well. They are made up of primarily of the glial fibrillary acidic protein. Okay, this is the point to remember: glial fibrillary acidic protein, because it is the main tumor markers for some main brain tumors. Okay, Now here is the example given. Here is an astrocyte in a special histochemical stain. Here is the nucleus and here is the cell process, and this yellowish appearance is because of this intermediate filament, which is the glial filament, made up of glial fibrillary acidic protein. Lamins have many isoforms in the nucleus. It is इसकी speciality यही है कि ये nucleus में होगा, ठीक है? क्यों? क्योंकि they form the nuclear lamina, which is the structural framework of the nucleus just beneath the nuclear envelope, ठीक है? They serve to support the nucleus of the and chromatin of a non-dividing cell. You can look in this image. Here is the nuclear membrane, here is the chromatin material, and here is the nuclear pore. And see, this network of intermediate filaments is called as a nuclear lamina. It is made up of these lamins.